Hello, this is Dr. Bates, and we're going to be talking about Chapter 19, Disorders Associated with the Immune System. In this chapter, we're going to talk about the concepts of how your immune system can respond to incorrect antigens. How can microscopic pollen from plants cause the problems we see in people today? We're going to talk about hypersensitivity, how these reactions occur, how they start the immune system. We're going to talk about immunodeficiencies, etc. So first, hypersensitivity. This is the response to antigens, we call them allergens, that lead to damage. Now, antigens, of course, means that is a molecule that antibodies bind to. We're going to talk about 4 different types of reactions. Type 1 or anaphylactic, type 2 cytotoxic, type 3 immune complex, and type 4 delayed cell mediated. Each type has a different time after exposure for the symptoms to occur. You do need to know this information. So type 1 is a quick response, is less than 30 minutes. Type 2 is 5 to 12 hours after exposure to the sensitized antigen. What this means is that the first time you're exposed to the antigen, it's a totally different story because your body's never seen it before. It hasn't made memory cells. It hasn't kept any of that information. So the first time is kind of free. It's the second time after you've been sensitized to that you're going to have problems. Okay, so we are talking about that second exposure. So type 3 is 3 to 8 hours and type 4 is more than a day. So first, type 1 or anaphylactic reactions. This is where IgE interacts with mast cells and basophils, causing them to undergo degranulation, aka they release their mediators that cause the reactions or initiate reactions in your immune system. So the antigen binds to 2 adjacent IgE antibodies, and the mast cells and basophils release histamine, leukotrienes, and prostaglandins. Each of these are going to cause processes that are going to cause the symptoms that we see. Histamine is going to increase the permeability of the blood capillaries. We get contraction of smooth muscles. We get an increase in mucus secretion. So all of those are going to be reactions to this process. Now again, this is going to occur minutes after a person that is sensitized to an antigen comes in contact with that antigen again. A picture showing the antigen binding to the antibody, causing the mast cell or the basophil to degranulate, to release all the other mediators that are going to cause the immune system responses that we see. So we have systemic and localized. Systemic is where it affects the whole system. It can result in circulatory collapse and death. This is when you're going to worry about how bad that allergic reaction is and when you use an EpiPen to overcome that. Could be a bee sting that could cause this response if you're highly allergic to it, aka if your immune system overreacts. Now, localized is where you have a specific spot. Localized is not throughout your whole body. So hives, hay fever, asthma, food allergies also fit into this. And it depends on your immune system response whether it would be systemic or localized, how bad that reaction would be. Now what we do to help you stop having this reaction is we can either take those items out of your diet if it's a food, out of interactions, say don't come in contact with, etc. But we can also desensitize you to that. That's where we actually give you the antigen in small doses so that your immune system will eventually decide that it should not react to this. We can cause the production of IgG so that antibody will act as a blocking agent for the reaction. Now, this can be done by hospital. We can also do this by presenting you with the antigen via honey. That's one way why people talk about taking honey. Now, it has to be local honey because that's the only one that would actually have antigens. And this is if you're allergic to pollen. It would be a way for you to desensitize yourself to it. Now, the problem, of course, is if you have a really bad reaction, then you could have that bad reaction to the honey. One of the reasons they say not to give children honey or nuts as a child, peanuts. That was one of the things, is that they said, keep children from eating peanuts when they're younger for fear that they're going to have a reaction. The problem is science has now determined, several studies have shown, that by stopping them from coming into contact with the antigen, they actually have a worse reaction. They're more likely to be allergic to peanuts than if we would have actually had them come in contact with them young ages. So right now, under the supervision of a doctor, they're actually taking children who have that high possibility of being allergic to and actually having them come into contact with that at a younger age. There is the hygiene hypothesis, and that is that by limiting the exposure, we actually decrease immune tolerance and the ability to cope with harmless antigens, aka the more you restrict yourself, the more you protect yourself, the less likely that your immune system will have options or knowledge about the system and therefore can overreact more easily. How can microscopic pollen from plants cause acute discomfort? It has to do with interactions of the immune system. Okay, so now we're to type 2. So type 2 involves IgG or IgM antibodies and complement. Of course, complement activation causes damage, cell lysis, and damage by macrophages. This example is an ABO blood group. We're actually talking about damage to the cells themselves, to your blood cells. Now, we have the A, B, O, and then the AB blood types. Now, we've talked about these before. You should have heard about or understand how the blood types work, and that is that blood type A has the antigen, the molecule on the outside of the cell that says, I am blood type A. Blood type A also makes anti-B, so it makes an antibody that actually would destroy B blood cells. If you're blood type B, you make the name B on the outside of your cells and you produce anti-A. If you're AB, you make both of those names on the outside of the cell and you do not produce any antibodies. If you're O, you have nothing on the outside of your cells, you don't have a name, and you kill or hurt or produce the antibodies against A and B. So if I give someone who is blood type A, blood type B cells, their immune system will destroy every one of those cells because they make anti-B. Same thing for if you're B. If I give you A, same story. If you're O, I can't give you anything but blood type O. And of course, if you're AB, you can take everything. I want you to notice over here we have a frequency. So the blood types have a frequency among the population. Certain genetic backgrounds are going to have different kinds. Again, you're going to have A antigen on blood type A, B antigen on blood type B. AB is going to have both, and then blood type O has none. So if you are blood type O, then you can accept blood from only O. And then to further add in, we have our RH factor. So we have our RH plus and minus. That's where the doctor tells you have O positive, B negative, etc. That's a second name on the outside of the cell. And it is another part that is important. Now, there are over 30 substances or molecules on the surface of red blood cells. The combination of these would give multiple different blood types. If you are O minus, you can only get blood from O minus because you hate plus. So you make the antibody against plus. If you are O plus, you can get blood from both O minus and O plus. And the same all the way across, as we said before. If you're A, you can get blood from A, but if you're A minus, only A minus. If you're A plus, it can come from any A. Now, blood type was discovered in the early 1900s. And it was determined then that you could only get blood from someone who matched you, whatever that meant. They weren't quite sure at first. There has been blood transfusions happened all along history, but many times the person who received the blood transfusion would die and they didn't know why. And it was because their blood didn't match. The reason why we might see the differences in different genetic backgrounds is that if you're blood type B, you are more susceptible to the Black Plague, because the Black Plague looked like blood type B, and therefore, if you had blood type B, you would not attack it as quickly as someone who had blood type A. And then blood type A is susceptible to smallpox, so once again, we would see a shift in the population where smallpox outbreaks occurred. One of the things that's important about this is that plus and minus, that RH factor. Now, it's called RH factor because it was originally discovered in rhesus monkeys. And what's important about it is that it has to do with pregnancy. So a mother who is Rh minus, who has a baby that is Rh plus, will actually respond to the fetus and destroy it. This is because this is considered foreign. The immune system is attacking it. Now, when you're pregnant, your immune system is actually decreased on purpose by the system because the baby can be identified as foreign. If you are Rh plus, your baby can be either one. It's not going to be a problem. So what we do nowadays is if your significant other, the father, if the father is plus and you are minus, we automatically give you a shot that will decrease this response. Now the catch with this is, again, I told you the first time's free. So the first time the mother is pregnant, until those interactions occur, and that's normally going to be late in pregnancy, it's going to be when during delivery, especially during the places where mother's and baby's blood mix. So in case you didn't know, baby has its own circulatory system. Mother and baby's blood don't mix. The baby actually steals oxygen from the mother. The baby's blood is more attractive or attracts the oxygen better and will actually siphon it or steal it away from the mother's blood. If it's your first child, you may not have this trouble until your second child, because when that baby is delivered, that's when the response would be initiated, and therefore the initiation would be waiting for the second pregnancy. Maybe you were pregnant and something happened, the fetus never developed, you might not even know you were pregnant, and therefore you still have a first response that caused the reaction. These antibodies make the baby anemic, which is why it would destroy the fetus in the end. So the Rh, again, it's found on the RBC, so the plus is found on 85%, they think, of the population. If Rh plus blood is given to an Rh minus, then that stimulates the anti-Rh and will destroy those red blood cells. So hemolytic disease of the newborn is where, again, a mother that's minus and a baby that's positive would cause the anti-Rh antibodies, and the second Rh positive fetus would be damaged. Thrombocytopenic purpura is where we have a drug that interacts with a cell, in this case a platelet, and makes this hapten complex. And the hapten complex ends up being attacked by an antibody that recognizes that drug, that antigen. Therefore, actually damage your own cells because of the drug. You'll notice this in older individuals. Sometimes they'll take a new drug and you'll actually see what looks like bruising all over their skin. And that's where their platelets have been destroyed because of the drug binding with it, because of an antigenic interaction. Combines with drugs, antibody and complement destroys the platelets. A granulocytus, that's where we have another drug that causes the destruction of granulocytes. And we have hemolytic anemia. Again, drugs bond to interact with red blood cells and you destroy your own cells.